So to introduce Alan, so Alan is, we're very grateful to him, first of all, with his busy schedule to come and talk to us tonight. In solidarity to our mutual Scottish roots, I've worn my Wilson tartan tie, which should be appreciated, no doubt. And I've unashamedly plagiarised Alan's biography from his University of Birmingham website. So Alan's a professor of microbial genomics. He's been funded by the EU, the MRC, Royal Society, lots of prestigious funders. And he works all over the globe with all sorts of different international partners. And most recently, during the COVID-19 outbreak, Alan was instrumental in setting up the Milton Keynes Lighthouse Lab and then the Birmingham Turnkey Laboratory. So today he's going to talk to us about all sorts of wonderful things that he's been doing there. On a personal note, I think Alan was one of the, on Twitter, certainly one of the sole scientific reasoning voices that people could rely on. I know he received a lot of flack for it, but I know myself and a lot of other colleagues certainly appreciated his take on the pandemic, certainly during the early stages of it. So over to you, Alan. We're really looking forward to your talk.
Thank you very much, Philippe. I shall now do the horrible IT parts of sharing screens and so on. So yes, thank you for the very kind introduction, Philippe, and also thank you for the invitation to present this. It's a great honour, and especially nice to be presenting to the East Midlands Charter. I obviously have quite an affinity for the region, having spent 10 very enjoyable years at Nottingham Trent University. It was actually a tough decision to leave NTU to go to University of Birmingham, but yeah, so lovely to be presenting to you all. The brief I was given for the talk was really a bit of a ramble of my career, so I make apologies at the start to anyone that's come expecting some dazzling hardcore high fidelity science. It's a bit more of a kind of tour of my where I started and how I got to where I am now, and there will be some COVID in it, and there'll also be some bacteriology in it. So hopefully I hit the brief. So yeah, as Philippe said, I'm now at University of Birmingham at the IMI. I've been there for coming up for 4 years now, and for my sins recently was made the director of the institute, which is an interesting task, but made very easy by very excellent colleagues. So I thought I would start with the very start of my pre-career really, because it's something that people don't tend to talk about a lot, I think, and particularly enough. High-profile academics don't talk about it a lot or enough. But I grew up in a town just west of Glasgow called Linwood. I went to this school here, Linwood High School, which is now demolished. This will be a bit of a theme of my talk, actually. And this is the town of Linwood. This is Linwood Shopping Centre, as it once was. It's now been demolished and rejuvenated, but it's infamous for having won the Architectural Carbuncle Award for the ugliest town in Scotland. I spent most of my time in this town, the neighbouring town of Paisley. And I spent most of my time there because I used to go and, basically I spent most of my youth at St Mirren Park watching my beloved St Mirren. And that probably set me up for a scientific career, given that I was exceptionally well adjusted to handling failure and handling rejection. Being a St Mirren fan, I think that's definitely what set me up for my career. I was lucky enough to get a place at Glasgow University as what we now rather endearingly refer to as a widening participation student, a term I truly hate, but I still consider myself very lucky to have been offered a place at Glasgow University, and I spent 4 of the best years of my life there studying microbiology, one of the few places that still does a proper microbiology degree. And it was just, it changed my life forever, and opened my eyes and opened my horizons, and it was a wonderful experience. Great university to go to, and some amazing teachers as well. This fellow here, Tim Mitchell, who ended up actually a colleague at Birmingham with me, but he taught me as an undergrad and was really the person that fired my interest in becoming a microbiological researcher. He used to give amazing lectures where he would basically break down a research paper for us and allow us to understand the, the design of the experiments and the reasoning behind the experiments. He's just a fantastic lecturer. And also Rob Aitken here, who was another fantastic lecturer, and really, really lucky to have been taught by these people. These, just completely inspired me that I wanted a career in science. And the other thing that did that was I did a work placement year during my degree at SmithKline Beecham, which no longer exists, obviously. It's now GlaxoSmithKline. I did the placement here. This is SmithKline Beecham R&D facility at a place called Frith House in Welland in Hertfordshire, which has now been bulldozed and is now a housing estate. And the year after I finished, SmithKline Beecham were aggressively bought over by Glaxo Wellcome to become GlaxoSmithKline. So that's two things now that I've been responsible for the destruction of SmithKline Beecham and Frith House and my secondary school. I'm sure it's just coincidence. When I graduated from Glasgow, again, extremely lucky. Luck's got a lot to do with things in science. I was incredibly lucky to secure a PhD position at University of Edinburgh with this chap here, Professor David Galley. And I learned so much in that 3 years that I was with Dave. It was an amazing lab, really, really wonderful lab. David just secured a veterinary teaching and research fellowship with DEFRA. And so the lab was awash with money and awash with very talented people. Dave Smith here was my second supervisor, and the two postdocs who I worked alongside in the lab were Andy Rowe, who's now a professor at Glasgow himself, and Nick Holden, who's now a professor at the James Hutton Institute in Dundee. And just working alongside these people every day was I was like a sponge for new knowledge. It was wonderful, really excellent experience again. And of course, by that time, you're completely hooked, right? And that's what you want to do. It's funny nowadays, obviously I have PhD students of my own, and I also examine a lot of PhDs. And one of the things that's always really quite surprising me still is the expectation level nowadays on PhD students. So this one paper in the Journal of Infection and Immunity in 2001 was the output, the entirety of my PhD, essentially in one paper. And I was incredibly proud of it. So I did my PhD on E. coli 157, and at the time, my supervisor David was very interested in, rather than studying an organism, you take one strain of an organism, the workhorse strain, and you understand it to the nth degree. It was David that really instilled this thing in me about studying bacteria as a population, because no two strains, no two cells will do the same thing within a population. And that was something Dave had really made his career on, with the type 1 fimbrian Fim switch and phase variation and so on. And so David really was the inspiration behind the PhD project, which was to look at lots of strains of E. coli 157 and basically just look and see did they all behave the same. And I was very lucky that they didn't. And essentially my entire PhD revolved around showing that if you took X number of strains of E. coli 157, they would all greatly differ in their ability to produce the key virulence factors, the type 3 secretion system, and to vary, to hugely varying extents as well. So this was pretty much my entire PhD summed up in one paper in infection and immunity. And of course, nowadays in the age of ref and so on, this probably wouldn't even be a paper that would be submitted to ref. And yet there you are. This was my PhD summed up, and it was such a thrill to have it published because it was an American journal, and everyone kept saying that if you wanted to be anyone in microbiology, you had to publish in one of the ESM journals. And yeah, that's such changed days now. I think PhD students, the expectation on them to publish 2-3 papers of very high quality, very, very different. I think I don't envy you. After my PhD, I ventured into the world of postdoctoral researcher, and it seemed a kind of natural move for me, having worked with David on a veterinary pathogen, a zoonotic pathogen, that I took a postdoc at what was the Veterinary Laboratories Agency, the VLA, in Weybridge in Surrey. It's now called the APHA. It hasn't been demolished, it's just changed name. And I did two really just so thoroughly enjoyable postdocs while I was down there. The first one I did with Professor Diane Newell and Gina Manning, and that was working on population biology of Yersinia enterocolytica, which was a really understudied zoonotic pathogen at the time, and Diane secured funding from DEFRA in collaboration with Brendan Wren to do a big population biology study of Yersinia and understand its true zoonotic risk in the UK. And that was a really enjoyable postdoc, loved it. And then when that postdoc finished, I decided to stay at the vet labs because this chap here, Professor Ian Brown, had just been made head of the avian virology lab at the vet labs and was looking for a postdoc to work on H5N1, avian influenza. Ian was convinced this was going to be the next big thing. And 3 months into my postdoc, the H5N1, I'm going to call it the avian pandemic, essentially. The virus swept around the world in wild birds and came into Europe and caused an enormous public health scare. And I was quite lucky that that happened 3 months into my postdoc with Ian. So I ended up immersed in the world of pandemic testing and genomics and how little did I know that would set me up for something that happened 20 odd years later. But that was a wonderful experience working on avian influenza right in the middle of the avian influenza bang, if you like. So it was great. Thoroughly enjoyed it. I think I think you'd be surprised what a big paper is. Again, I've mentioned the world of ref that we all live in nowadays in universities, and this push for science, Cell, Nature papers. These are two papers that came from my postdoctoral research at the vet labs. This is one from the project I did with Diane, and it was basically what we did was we took the UK abattoir survey. So every few years there's a big UK abattoir survey. And what we did was we got access to all of the Yersinia enterocolytica that had been isolated during that study, and we basically catalogued them, and we made a catalogue of all the biotypes and serotypes and genotypes present of Yersinia enterocolytica in cattle, sheep and pig in the United Kingdom. And we published it in Letters in Applied Microbiology, which has an impact factor of around about 1.52, I think. This is my third most cited paper. And this one here, which I'm in the middle of, was one that came out of my time with Ian's lab. And this was basically, as I said, 3 months in, the avian influenza pandemic in wild birds began and came into Europe. And so Ian, our lab was also the European reference lab for avian influenza. And so what we had to do was develop a real-time RT-PCR assay that would basically be validated and function as the accepted diagnostic test for avian influenza in wild birds and poultry. And we did that and we published it. My role in it was Marek actually designed the assay. My job was to infect, it sounds rather barbaric, infect lots of ducks, turkeys and chickens with H5N1 and then ensure that the PCR worked in all different types of tissues and determine a limit of detection and so on. This paper was published in the journal Avian Diseases, which again has an impact factor around about 1.6, I think. And this is my second most cited paper of all time. And arguably, given the last 12 months or so, it could well be described as the most important paper I've ever been involved in, because it's really due to this work that I ended up being so heavily involved in COVID. So you'll be surprised, I put the headline there, you'll be surprised what is a big paper. A big paper isn't necessarily a paper in Science, Nature or Cell. Sometimes it's the things you think are little throwaway papers that end up having the biggest impact. And yeah, so that's a bit of a very brief whistle stop of the outputs from my postdoctoral time. So after those two postdocs, I was kind of, as you do when you're a postdoc, you start to get delusions of grandeur, and you start to get ideas above your station. And I decided that my ideas were much better than Ian Brown's and Diane's, and that I wanted to be an independent researcher. And started to apply for a number of jobs, got a few interviews, got a couple of offers, but got an offer from Nottingham Trent University, originally actually to go there on a teaching only post for 2 years, kind of try me out, see if I was any good type post. And I took it and moved up to Nottingham. And of course, when you start as a PI, you're absolutely terrified because you think, well, what am I going to work on? I knew I couldn't work on H5N1 because of the containment level, because of the fact it was a controlled pathogen. I knew I wasn't going to continue working on that. And actually, I wanted to go back to working on bacteria, which are much more fun to work on anyway. Apologies to any virologists. So I mentioned this chap here, Brendan Wren. Brendan was the co-investigator on the first postdoc that I did with Diane Newell on Yersinia. And Brendan is at London School of Hygiene and just an amazing character, a wonderful scientist, and just a really, really nice chap. He really turned into quite a friend, colleague, and mentor for me. And Brendan introduced me to this chap here, Nick Thompson, who at the time was had just become a senior scientist at the Wellcome Trust Sanger Institute in Cambridge. And Nick was really driving the idea of using the genomic capacity at the Sanger Institute to go beyond sequencing reference strains and sequencing populations, which was right up my boat, given the sort of the things that David Galley had drummed into me about studying bacteria at a population level. So Brendan introduced me to Nick and gave me wonderful mentorships. They still do give me wonderful mentorship and advice, and these two guys really got me into genomics. I'd done a little bit of genome sequencing and phylogenetics during my postdoc on avian influenza, because we had to sequence the H gene of all isolates and make a phylogenetic tree to position the strains and look at transmission and spread of the virus. But it was really Nick and Brendan that got me into genomics. And when I was starting at NTU, the high throughput sequencing with Illumina technology and so on was really just taking off, and Nick convinced me that this was where I should focus my efforts and learn some genomics and get into doing genomics, and obviously wonderful advice. It worked for me anyway. It turned out to be wonderful advice. So this was my group, NTU, and we started very small. This is, I don't look like that anymore, I can guarantee it. This is my first ever group. So I started with this young lady here, Sandra Reuter, who's now an independent research fellow back in our home country of Germany, in Freiburg. And Sandra came over to study a PhD on Yersinia and doing Yersinia genome sequencing with Nick and Brendan. And that was a PhD that was very generously funded by Nottingham Trent to get me up and running. We secured some funding from the Dowager Countess Eleanor Peel Trust, which funded a PhD studentship to work on uropathogenic E. coli. So I decided to go back to the two things. I'd studied E. coli and Yersinia. And this is Gemma Clark, who was a graduate of NTU and took that PhD position with me to work on uropathogenic E. coli. So we had Sandra, who was going to do genomics of Yersinia, and we had Gemma, who was going to do genomics of E. coli. So I was wonderfully happy. And this lady here is Jenny Inns. She got a job as a postdoc on a project that was funded by the Technology Strategy Board, now Innovate UK, to develop rapid diagnostics for intestinal pathogens. So this was the group, and then we also got some funding from FP7. I was involved in a large EU FP7 grant, also developing rapid diagnostics, and a couple of grants. 2 PhD students, a postdoc. I thought this was the perfect group. I still think that's actually pretty much close to the perfect size of group, but it was really fun. I enjoyed it so much. Sandra's super talented, as I said, is now a research fellow in Germany. Gemma's now basically running virology in the hospital in Nottingham. She's a consultant clinical scientist in virology at QMC. It was great. It was really great start to the lab, and the lab kind of grew a little bit over time. Lots of PhD students came and went, and it was started small, very focused, and at the time it was all about trying to make a name for myself in those 2 areas that I decided to focus on genomics of Yersinia and genomics of E. coli, and particularly uropathogenic E. coli. you know, the beauty of being small but focused is that you don't have to worry so much about the massive labs out there that are obviously going to outcompete you if they want to. You know, it's kind of finding your own little niche and then just doing that and nothing else. And that really worked for me. And, you know, I've just put up a couple of examples here of stuff that we found. This was the output from, you know, I mentioned about how different PhDs are now. I have one paper from infection immunity. This is the output from Sandra's PhD, which was a paper in PNAS, where essentially what Sandra did was, along with Nick Thompson, was we sequenced representative strains from every single species in the genus Yersinia, which was something that hadn't been done before. No one had ever sequenced an entire bacterial genus. And Nick was convinced that we would find something really cool if we did this. He wasn't sure what we would find, but he was convinced we'd find something really cool. And sometimes it's good not to go with a hypothesis. But by sequencing every single species in the genus, we found something truly remarkable, which was that the human pathogenic Yersinia pseudotuberculosis and Yersinia pestis, which is a subclone of pseudotuberculosis, and the other human intestinal pathogenic Yersinia enterocolytica, are at diametrically opposite ends of the genus phylogeny. So those two species are as distantly related as they can be in the genus, and yet they're the two human pathogenic species. And what was even more remarkable was that they'd actually evolved pathogenicity in an exactly parallel fashion. So a beautiful example of convergent evolution. The same virulence plasmid had been acquired by pseudo-TB and by enterocolytica. And then there'd been this process of genome decay to go from mildly pathogenic pseudotuberculosis to highly pathogenic Yersinia pestis, and to go from the low pathogenic Yersinia enterocolyticus to the high pathogenic Yersinia enterocolyticus, the exact same process of genome decay. So independent capture of a virulence plasmid and convergent evolution of pathogenesis by streamlining of the genome and becoming more, you know, kind of metabolically less flexible. And, you know, I think I'm right in saying it was the first real example of this parallel evolution of pathogenicity in a bacterial genus. I'm hugely proud of that work. Gemma's work was around uropathogenic E. coli. We got into a bit of hot water here because we did come up against a huge multinational consortium. But essentially, Gemma had fallen upon the fact that the isolates, the urine and bloodstream isolates we were collecting from QMC in Nottingham, there was an overabundance of this particular E. coli ST131, which was being described as this multi-drug resistant pandemic E. coli. And sure enough, we found that it was circulating quite large numbers in the hospital in Nottingham, hugely drug resistant and very clonal. So very small numbers of strains we had, but from our collection, we showed that E. coli ST131 was this really, really highly clonal organism that was carrying ESBL plasmids and was causing huge clinical problems. And Gemma's paper in JAC was actually the very first E. coli ST131 genome paper published. After that, there came some really big ones in PNAS and in MBio and so on. But Gemma managed to get her 12 genomes out first, which she was very, very proud of. We also published a paper in Emerging Infectious Diseases describing this rise in multi-drug resistant E. coli and bacteremia infections, again, working with Viv Weston and Matt Diggle at the QMC in Nottingham, and you know, kind of amongst the first groups to really describe this worrying increase in multi-drug resistant E. coli and bloodstream infections, which of course is now well known, and there are government targets to try and combat that. But again, you know, sort of at the vanguard of that. And then I really started to branch into sort of hardcore evolutionary genomics as well, with the help of a wonderful collaborator, Yuka Karanda. And this is a paper that we published in Genome Biology Evolution, which was showing that the evolution of these E. coli ST131s, these highly drug-resistant clone, was actually marked by, it was clonal, and there was a reduction in recombination, and basically this bug had stopped sharing its genetic material with other E. coli and was becoming this streamline pathogen. So that's kind of a bit of a snapshot of what I what my group delivered at NTU. I say wasn't me at all. It was just really some phenomenal people that came and worked in the lab with us, and very lucky to have had such talented people come through the lab while I was at NTU. So this is kind of my timeline at NTU. I started there at 2006 as a lecturer. Lots of teaching, but you know, the clue's in the title of the job, right? As a lecturer. Just didn't complain, got on with it, got a couple of PhDs, my postdoc, couple of grants. That sets you on the way with your research. A couple of years later, I was made a senior lecturer. Teaching load goes down because you've got some grants. Lab starts to grow a little bit. And then by 2012, I was a reader in microbial genomics, and my teaching had really dropped down, and the lab was getting bigger and we were getting more grants and so on. So it was a great time. I really enjoyed myself at NTU. And I would never have anything bad said against it as a university. It set me on my path, and I had a wonderful time. There's some great colleagues as well. I noticed one or two are in the audience, and I apologise if you've seen this talk before. You might have done, I'm not sure. The one big challenge I had at Nottingham Trent, I think it's fair to say, was we were getting grants. I was getting EU grants, charity grants, Technology Strategy Board grants, collaborating on larger grants, but I was finding it really hard to secure, you know, UKRI, or you know, as it's now called, the UKRI funding as the principal investigator. And at the time, I thought it was fine. It was something I could live with. I was really enjoying my time at NTU, and you know, I was publishing in PNAS and so on, and I was loving it. And why should I consider it an issue? But then I went to it was called the SGM, of course now it's the Microbiology Society. I went to the Microbiology Society annual conference in 2015, which was in Birmingham. And this fellow here, Mike Prentice, is an old friend and collaborator of mine. We worked on the Yersinia stuff together, and Mike was at Cork. And I bumped into Mike at the conference, and he said, have you seen that Birmingham are advertising for, I think they were advertising for 6 lectureships in microbiology, which was kind of unheard of at the time. And Mike said, you should apply. And I said, no, I'm not going to apply to Birmingham. I'm really happy at Nottingham. And anyway, they'll be, you know, they're out my league. I won't get a job there. They'll be looking for someone at some hot shot from somewhere or other. But of course, Birmingham had, you know, the IMI had just been set up by Mark Palin and Laura Piddock and Ian Henderson, and it looked like a place that was really going places in terms of microbiology. So Mike said, you should apply. And then Mike said, I tell you what, I know 2 people at Birmingham really well. Let me introduce you to them. So he introduced me to Nick Lohman, who I knew of, you know, the microbial genomics whiz kid, who was at Birmingham and was doing great things in genomics at Birmingham. And this terribly intimidating looking chap here, who's actually one of the loveliest men you'll ever meet, Ian Henderson, who was the director of the IMI at the time. So Mike introduced me to Ian and Nick, and we got chatting about IMI and Birmingham and microbial genomics, and should I apply for the post? And Ian said, well, why don't you apply? You never know if you don't apply. So I applied for a lectureship at University of Birmingham, and lo and behold, I ended up moving there, and I moved there in September 2016. And I think it was the biggest thing I've done in my career, and probably in my life as well, because what it meant was we ended up as a family leaving Nottingham, a city that I loved living in, and relocating to the West Midlands, and it felt like the biggest, scariest thing I'd ever done in my life. But I just felt it was a move I had to try and have a go at. So I moved to Birmingham. This is the view when you come out the Biosciences building. You're looking at Old Joe here. I was given anyone who's been in Birmingham Uni will know the Biosciences building is, well, it's not the nicest. It's probably ready for demolition in the near future, hopefully. But yeah, you know, I got a kind of crumbling office and set myself up with every computer I could find, and set myself up as a one-man genomics research band at Birmingham. None of the people that were at NTU moved with me. I had a couple of PhD students who were coming to the end of the projects. It was better for them to stay at NTU, so I moved across myself. So it was me. I had one grant I moved with me, a Royal Society grant, and I got a research assistant for my startup, and off I went. And it's been a roller coaster of 4 years. It's been wonderful. But the thing about IMI in Birmingham really is it's just an incredible environment with phenomenal people. I've already mentioned Nick. I mean, he is I would challenge anyone to name me a brighter or better genomic scientist than Nick Lohman. He's incredible. And just being able to meet Nick every day, bounce ideas off him, you know, chat about analyses, chat, am I interpreting this correctly? The office next door to me, this chap here, Willem van Schaik's in the office next door, again, one of the brightest antimicrobial resistance brains I've ever met. It's just wonderful being able to collaborate with them. You know, we've got Birmingham's renowned for its antimicrobial resistance research, the strength of that, developed for years by Laura Piddock, and then people up and coming stars that came up through Laura's lab, such as Jess Blair. It's just a great environment, and you know, I'm lucky enough to again start to build my own group in Birmingham, and great PhD students, wonderful postdocs, Emma and Stephen, and just a really nice environment. And I think about the environment for me, it was something that Brendan Wren said to me when I took the job at Nottingham Trent, and you know, I said to him, so what's the most important thing you could tell me? He said, well, you always do the best science with a smile on your face, so make sure you've got a smile on your face, and all the good science will come from that. And I think he's right, and it's something I've always tried to do, is make sure that the people who come and work in the lab enjoy it, and really enjoy it, and they do have a smile on their face, because invariably you do your best science with a smile on your face. So my Birmingham timeline so far is, as I said, 2016, I moved there on my own with one grant, and then a year later I was promoted to reader on the back of a couple of big grants and a couple of nice papers, and that allowed the group to grow again because of the grants. And then, as I said, in 2019 I was promoted to professor, and I was asked if I would be the director of the institute when Ian Henderson took the offer of a lifetime and moved to University of Queensland in Australia. And you know, the grants continued to come, the groups continued to grow. We've had funding from Wellcome BBSRC, MRC, Royal Society, and NERP grant most recently, and it's just luck. You know, I please don't think that I'm being braggadocio here and anything like that. It's not at all. I know this could dry up tomorrow. It's riding a wave of a topic that's considered important and sexy at the moment, and you just have to ride that wave for as long as you can until it crashes down and the next big thing comes along. But I've really enjoyed the 4 years. As I say, it's been a bit of a roller coaster, and yeah, I'll enjoy it as long as it lasts. But I am realistic that what's the saying, that today's headlines are tomorrow's fish supper wrapper. So I'll enjoy it while I can. I think I decided here a little bit of science. I'm not going to get too bogged down in this. I know it's a very diverse and wide-ranging audience, but just to give you a bit of a flavour of some of the stuff that we've done in the time I've been at Birmingham, I've given a couple of examples here. I think we've continued the work on E. coli ST131, this multi-drug resistant pandemic clone of E. coli. I think it's a fascinating bug, and you know, we've kind of got into the large genome numbers on this bug now, and the last paper we had was 2,000 ST131 genomes. But it's a really fascinating bacterium because you have this clade C here. This is all the multi-drug resistant strains. So every one of these has a multi-drug resistant plasmid and has come from a bloodstream or urinary tract infection. And this is this globally disseminated pandemic clone, very little diversity in it at all. And the really interesting thing about this is that it's evolved from a completely drug susceptible background strain. So this clade A, and this clade A very rarely causes, well, we thought very rarely causes disease, never carries drug resistant plasmids. And so actually it's a perfect model for studying the evolution of antimicrobial resistance, because you can track the changes in the lineage as you go from the drug susceptible non-pathogenic clade A to this sort of intermediate, slightly drug resistant, sometimes causes disease clade B, to this highly clonal, highly drug resistant, highly pathogenic clade C. And that's really what my group, a lot of the focus in my group has been over the last 5 or so years, has been how is E. coli ST131 gone from here to here, and what are the processes that have driven that. And I think in doing that, we've also taken a different approach to how to study bacterial genomics, in that lots of bacterial genomics certainly not now, but a few years ago, was most bacterial genomics was around what we call core genome stuff. So you take genes that are present in every strain, and then you look at the mutations in those genes, and you make a phylogenetic tree, and you look at those mutations, and that tells you everything you need to know. But in a species like E. coli, genomically, genetically, is incredibly weird, because it has this what's called a huge pangenome. And I think this is one of the most exciting things in bacterial genomics, this idea of a pangenome, is that any two strains will have completely different genetic content, totally different sets of genes. And up until a few years ago, people never studied this pangenome, all these different genes that differ really. Most studies were in this, they were core genetics and core phylogenetics. And we took a different approach to studying ST131 from some of the other bigger international consortia, and we studied that accessory genome, so the genes that differ between strains. We also looked at things like promoter regions. No one ever looks at the promoter regions when they're doing genomics. They just look at the genes and differences in the genes. And we thought, well, differences in promoter regions are going to be really important, right? They could drive more biology than small genetic changes in genes. And that was very much that drove our analysis for a couple of years. And we were fortunate enough that it was a good call, by studying the accessory genome and the regulatory regions. We completely changed our understanding of how ST131, this multi-drug resistant clone, had evolved from its background neighbours. And essentially what we found was it was all to do with how plasmids and plasmids, but there's no fitness cost. It tweaks things to get rid of the fitness cost. And yeah, that's something that we've been very proud of. We followed that on by, again, with E. coli ST131, was looking at more detail at this pangenome idea and genes that differed between strains. And again, by comparing the drug-resistant clade C with the drug-susceptible background A's and B's. And what we wanted to do is we wanted to say, okay, what's different about the drug-resistant ones from the drug-susceptible ancestors? And by looking, rather than looking at, again, core gene SNPs and so on, we looked at the genes that differ in content. And essentially what we found in this paper in MBio was that if we looked at that, what we found, each of these little blue blocks here represents a gene that's involved in metabolism, and primarily anaerobic metabolism. So that's the ability to essentially survive and metabolize successfully in the intestinal, the mammalian gastrointestinal tract. And what we found was that there's huge diversity in anaerobic metabolism genes in the drug-resistant strains that isn't present in the ancestral drug-susceptible strains. And what this is it's an enormous signature of selection going on in the drug-resistant strains. And so essentially what's happening is, rather than selection on a gene, there's selection on a process. So it's what we call negative frequency dependent selection. It's selection on the process of being able to metabolize in the intestinal gut. And that is what really makes the drug-resistant ST131 different from its drug-susceptible ancestor. It's essentially evolved to be a better metabolizer in that harsh intestinal environment. And so, kind of changing how we think about the evolution of drug-resistant clones of E. coli, it's not really all about the drug-resistant plasmids. There are lots of other processes that underpin the evolution and emergence of these things. Most recently, a paper we've had out was trying to understand then. So if drug-resistant E. coli are better at colonizing the gastrointestinal tract, do you see that in real life? And quite a few studies had shown that if you travel to South Asia or Southeast Asia, it's highly likely that you'll be colonized by a drug-resistant E. coli. And so we decided to study this in real time, and working with Anu Kantala and Paul Newton and Laos and Esther Kunsley. Basically, Anu and Esther ran a 2-week training camp in Laos for Swiss medical students, 35 Swiss medical students. So they all went out to Laos for 2 weeks and did some training on antimicrobial resistance in Laos. And as part of the trip, they agreed to provide fecal samples as and when they produced them, such that we could culture them for multi-drug resistant E. coli. So they were all the fecal samples pre-travel were negative for drug-resistant E. coli, multi-drug resistant E. coli. And then every time they had a bowel movement, we took the feces and we cultured it. And every single participant in the course was colonised by a drug-resistant E. coli within 2 or 3 days of arriving in Laos. And so this really proved to us that what we were suggesting was true, that drug-resistant E. coli are better at colonising your gastrointestinal tract. They are superb at it. And what we saw actually was competition between different drug-resistant E. coli. So here there's a participant colonised by one strain, then a different strain comes in, and then a third strain comes in and kicks those out. And you can see that here as well. So a really dynamic process. But I think the beauty of what this paper showed is that this is what happens. This is what makes these multi-drug resistant E. coli different. They're wonderful at colonising your gut, and everything then comes from that. And very recently this week, we've had a follow-up paper on that with ST131 and studying ST131 in Norway. And you can see here how ST131 over time became the completely dominant E. coli strain type in clinical samples in Norway. This is bloodstream infections. But what was really interesting here was it wasn't the multi-drug resistant clade that was dominating. It was actually the drug susceptible one, the clade A. And that's because of very different antimicrobial prescribing in Norway. But what was really important here is this showed us that it's not drug resistance that made ST131 successful. It's the inherent ability to colonize the gut better. And then the multi-drug resistance kicked in. And in Norway, this data set shows us that beautifully. So what we think, I think very whistle-stop tour of 4 papers there, but what I think we've started to do in Birmingham is, in the group in Birmingham, is start to rethink how we understand the evolution of antimicrobial resistance and antimicrobial resistance clones, particularly multi-drug resistant ones. And really, this is now our working hypothesis for how multi-drug resistant E. coli evolve, and this is something that we are now going to be pursuing for the next few years in our lab. And there are 2 key things. The first one is this idea of when a drug-resistant plasmid enters an E. coli, one of 2 things can happen. So when the plasmid comes in, lots of genes on the plasmid, which means that the transcription and translational machinery, which is required to transcribe and translate genes on the chromosome, that machinery is going to be recruited to the plasmid, and you get a fitness cost, because that means you don't increase the amount of transcription and translational machinery you produce. What happens is it's kind of sequestered and quenched by the plasmid, and so you get a drop in transcription translation of the chromosome, and you get a reduction in fitness. And so one of 2 things can happen. In most E. coli, the plasmid will be destroyed, or if there's a toxin antitoxin system and the plasmid can't be destroyed, the bacteria containing the plasmid will die because they are not as fit. What we think happens in successful multi-drug resistant clones is when the plasmid comes in and starts to hijack the transcription and translation machinery, you get adaptive mutations in promoter regions, such that this maneuver, this reshifting of transcription and translational machinery happens at a reduced cost. And so these E. coli basically can handle this happening better, and there's no fitness cost, and they thrive. And what we think drives this is this ability to metabolize better in the gut. So something like an E. coli ST131 has evolved by being able to induce the production of weird metabolites. This is often by inflammation. We know this in Salmonella, but basically induce the production of weird and wonderful metabolites in the gut that the commensal E. coli cannot metabolize. The incoming E. coli invading drug-resistant E. coli can. That gives it a competitive advantage, and that allows it to colonize the gut. So kick out the commensal E. coli and displace it with this invading drug-resistant one. And of course, the longer you hang around in a mammalian intestinal tract, the more exposed you're going to be to antibiotics, and the more selection there's going to be for this process here of acquiring a drug-resistant plasmid. So that's our big hypothesis now, and that's going to be the focus of, I think, the next 5 years or so of the work going on in our lab. So I was just going to sort of coming towards the end now. Hopefully I'm not waffling. If I am, then Philippe needs to feel free to come in and shepherd's crook me off. But I think something I want to point out, I've heard this a lot of times since I became institute director, is that at some stage as an academic and as you get older and not so wiser, you have to choose between research and admin management. And I noticed that Nigel Brown's in the audience here, so as one of the most eminent senior academics in the country. I'd be interested to see if he agrees with me on this. But you know, there is this dogma that if you take a head of school, head of institute role, your research is going to die. You have to choose between the two. I never really felt that, and the advice I give to anyone now who asks me is keep all your options open and be open to all the opportunities. When I was asked if I wanted to be institute director, I thought I have to do this, and not for any sort of career-driven aspirational goal. It was, let's give it a go. Let's see if I'm any good at it. And you know, you never know. It might be I want to do rather than write lots of grants and get rejections all the time. Maybe I want to do this. So I did it, and being an institute director has not gotten in the way of my research. We still have a very active research group. We're still writing grants, writing papers, getting grants. So it hasn't gotten in my way. I think you need a good admin team around you, undoubtedly, to help you with the work burden. But my advice really is don't be this blinkered. I don't think you have to, I don't think it's one or the other, and I think you can do both. And the good thing about taking on leadership roles is they can present opportunities. So as you all know, last year I was asked to go and help set up the COVID testing lighthouse lab in Milton Keynes. That didn't really come about because I was the country's best virologist, right? I hadn't done virology for a long time. I've been involved in developing the H5N1 diagnostic, and the people I was chatting to knew I'd done that. And they knew I'd worked in an H5N1 diagnostic lab. But really, the invitation to do this came because I'd just become the director of the IMI, which is one of the biggest groupings of microbiology and microbiologists in the country. You know, there are lots more perfectly better qualified virologists in the country could have been asked to do that. This came from the fact that I had this, you know, sort of visible leadership position. So undoubtedly that's why I was asked to do that. And it was incredible. It was a really wonderful and phenomenal few months. You know, it was, the lab's based at the National Biosample Centre in Milton Keynes. I arrived there at the end of March to a very small lab with 6 MSc2 cabinets to do the work in, and about 12 volunteers, and we were doing everything manually. And a month later, we'd moved into this enormous, you know, football pitch-sized lab with lots of automation. There were 14 MSc2 cabinets with manual processing of samples. We had these large cabinets which we could put robot platforms in to do automated processing of samples. So each of these hoods could do 96 samples an hour. These things could do 96 samples in around about 15, 20 minutes. An incredible setup, you know, lots of automated RNA extraction robots, Kingfisher robots, big rooms full of PCR machines. And what that allowed us to do was a ridiculous number of COVID testing samples. This was basically what allowed Pillar 2 to happen, the idea that you could test anyone in the country who felt they had COVID symptoms for the presence of SARS-CoV-2. And, you know, by the time I left, we'd done just under 1 million samples in the space of 3 months. And, you know, we used to, this is the sample receipting room. Around about 50,000 samples a day were coming into the Milton Keynes lab for processing. I've never done anything like it. I'll never do anything like it again. It was truly incredible. So like I say, very small beginnings, you know, a very small lab, 12 people, 6 cabinets. We started off doing around about 1,000 samples a day. Horrifying positivity rate, around about 30% of all the samples were positive. And it was slightly terrifying. You know, in a brand new lab none of these guys, they were all volunteers, none of them were being paid, they'd all volunteered, none of them had worked in a diagnostic lab, but some very talented people. This is Joanna Viana, who's a research fellow at Birmingham. This is Angus Best, who was working at the vet labs at the APHA. He was seconded. You know, just remarkable. And like I say, in the space of a month, we'd gone to this. This is the throughput. This is data from the LIMS system in the lab. So you can see here at the start of April. So each of these little marks is when a 96 well plate goes onto a PCR machine. So you can see at the start of April, we were doing, you know, maybe a dozen or so 96 well plates a day, most of them going on in the afternoon. So you start in the morning, they go on in the afternoon. And then within a month, we had gotten to this, and then when we went to 24/7 operation, you can see how that just ramped up significantly as the lab automation kicked in, the robotic platforms kicked in, and we went from 1,000 samples a day in a month, within a month, from 1,000 samples a day to 30 to 40,000 samples a day. Really incredible. And then, of course, you know, once the lab was set up come June, I felt my job was done, and I went back to Birmingham, only to be asked by the Department of Health if I could set up another lab at the University of Birmingham, with so-called Turnkey lab. And I know that Benitez and a couple of other people who joined us in Birmingham are here and on the call, and I can't thank them enough for what they did to make this lab reality. But essentially what we did was, in 2 weeks, we took this horrible lab space in the Birmingham Medical School building that wasn't really being used, and we converted it into a fit-for-purpose modern lab without parquet flooring, but actually proper lab sealed flooring and sealed benching. We took delivery of lots of reagents. We got some robotic platforms in to do the testing. We also got some MSC2 cabinets for manual testing. And at the end of, in September, we started to take receipt of our first samples. And so we started doing Pillar 2 testing at Birmingham as well. And one of the key things we had was, I've already mentioned this chap, Angus Best, who was a postdoc alongside me at the vet labs, and then by luck ended up being seconded to Milton Keynes the same time as me, and we convinced him that actually his future lay in Birmingham, and he had to come and run the Turnkey lab for us, and he did. And he ran it wonderfully. The success of that Birmingham lab is all down to Angus. But really, just, yeah, it feels quite a surreal experience now having done this. To have done it at Milton Keynes once was really weird, but to do it again at Birmingham was super weird. And you know, what we set up at Birmingham was essentially a miniature Milton Keynes. So we a modular lab system. So the first part was we decided to heat and activate the samples, just to make it safer to work with. You know, we're working in a university building, in a university lab, shared building, lots of other labs. And for extra security, we decided to heat and activate the samples. That was the very first step. That allowed us to unbag them, uncap them, put them on a robotic platform for sample prep. So basically, the sample gets pipetted into a plate containing lysis reagents, and then, you know, the sample is completely safe. Then it can go on to the Kingfishers for RNA extraction. And then they go on to this next robot to prepare the 96-well PCR plates, and then they go on to Quant Studio qPCR machines. And this was essentially the makeup of the lab. It was a mini Milton Keynes, a modular system, brilliant staff. Lots of people who'd been at Milton Keynes decided to come up and join us in Birmingham, and I can't thank them enough. And unfortunately, the government decided after 6 months that we were no longer needed, and those people have had to move on to other jobs, and which saddens me greatly. But I can't thank them enough for the work they put in. They were phenomenal. And I think one of the most exciting things about the time that we did in Birmingham with the COVID testing was, because we're in a university, you know, the thing that annoys me a little bit about Milton Keynes is it was really just testing. That was all it did. And there was scope for so many amazing publications from that lab. And I was adamant that when we got the lab up and running in Birmingham, it wasn't just going to be a testing lab. We were going to publish our findings and do some research. And this is one of them. I think this was the most exciting thing that happened in the 6 months we operated in Birmingham, was the discovery in early December that the PCR assay was starting to work differently. It's a 3-gene assay, and one of the gene targets, the S gene, had stopped amplifying in lots of positives. It was so-called S gene target failures. I'm working with Nick Lohman and Josh Quick, who run the COG, the genomics, COG COVID Genomics Consortium lab in Birmingham. We sequenced some of these, and what we found was that there was a deletion in the S gene, exactly where the fluorescent probe binds in the assay. That meant the S gene was amplifying, but you couldn't detect fluorescence. And this was happening in one lineage of the SARS-CoV-2 virus, the infamous lineage we now know as the Kent variant B117. And essentially, ourselves and Milton Keynes were the first labs to identify the S gene target mutation, and that it was in a specific lineage B117. And we were in part of the report to the Department of Health and the government about the rapid emergence of this S gene target failure, this B117. And what you can see here, the grey bars are absolute numbers per day. And the lines here are 5-day rolling averages of the percentage of our positives that were B117, the S gene target failures. And what you can see is over December, this really rapid exponential rise in the frequency of B117. We were testing samples from all over the country, 4,000 or 5,000 samples a day. At the peak of this, actually, Benita and the guys were doing about 6,000 samples a day. And you can see how quickly B117 swept the dominance to almost 90, 100% of all viruses we were detecting were B117. So something incredibly proud of the efforts that we put into the COVID testing, national COVID testing, and to be part of something as exciting as this is. I hope the guys who did work in the lab and who are sitting in the audience just now agree. One of the most exciting things I think any of us will ever do. So hopefully I have kept to the brief and I've kept to time, and hopefully it's been just the right level of science and waffle from me. There are a couple of people I have to mention. This slide could be thousands of names, but a couple of key people, Zhiyong Zong in Chengdu in China. I've been a collaborator and friend of Zong now for about 12 years. He's the reason we can do all the exciting stuff we do with the multi-drug resistant E. coli, because they all emerge in China, and Zong finds them long before we find them in Europe. And it's Zong that gives me emails and phone calls and says, Alan, we've got another one. We've got another weird clone. Shall we study it? He's an amazing friend and colleague, and he does incredible things with not much resource, just an absolute genius of man. Yuka Karanda has taught me so much about bacterial evolution and genomic analysis, and one of my best friends and best colleagues. I was struggling with who to mention him from NTU, but I've picked Ellen Billett, who was just such a support to me research-wise when I was at NTU, a wonderful mentor and really supportive, really encouraging, just a great colleague, and I miss working with her regularly. Some great conversations and verbal battles with her at times as well. And my current boss, David Adams, my head of college, again, just a really supportive person who refuses to listen to my my imposter syndrome moans, and gives me the belief that I can do things when he asks me to do them. And he's a wonderful boss. Everyone who's worked in the lab, too many to mention, past and present. I've been so fortunate to have some of the most talented PhD students and postdocs I could imagine. And they're the people that produce the research and the quality of the research. The entire Lighthouse Lab team at Milton Keynes, they were incredible. 240 people in 4 shifts, working 24 hours a day, 7 days a week. Most of them volunteers. Just I will never experience anything like that again as long as I live. And then the guys that came to Birmingham and set up the Turnkey Lab, they were they were as heroic as well, and I hope they enjoyed their time in the lab. I certainly enjoyed working with them, because they are really just wonderful colleagues, great colleagues. So with that, I think I shall stop sharing my screen. I hope you enjoyed it, and I'll see if there are any questions from you. Thanks very much.